It's a good pleasure to speak to you today about AI and data curation. And I think I slightly modified my presentation title, and because I think a lot of you were probably interested in seeing AI for assisting data curation. But I'd like also tell you about my perspective on the other side. I think data curated data is really valuable for today's AI and large language model research. So before I do that, just want to do a quick introduction. My name is Zhiyong Lu. I come to you from National Library of Medicine. We are part of the NIH Intramural Research Program, where I lead a research group interested in using AI and machine learning for biomedical text as well as image processing. So on a routine basis, we primarily work with biomedical text data that includes tens of millions of PubMed abstracts, as well as millions of full-length articles in PubMed Central. When access is given, we also directly working on patient data, including electronic medical records, as well and radiology.
Breaking up a little bit.
I am.
Or unless you stop talking for a few seconds.
Okay. Well, let me know if that continues. I hope not.
You're good now. You're good now.
Okay. Because I could switch a different computer, but that will have some interruptions. So let me know if that problem continues. Okay. So because of the topic today, of the audience today, I'm going to talk mainly focusing on the text side. And we'll try to cover some of the recent studies that we published from both, using AI for data curation and vice versa. But before we get into the details of these studies, I would like to starting with PubMed, because this is a database that perhaps everybody in the audience also contributes to. You publish a paper, you deposit your paper into this database. As of now, we have now over 38 million articles in PubMed, title and abstract. On an average day, we have about 2.5 million users doing about 3 million searches and 9 million page views. That traffic compares nothing to Google searchers, but does make PubMed one of the top 3 or top 5 most accessed federal government websites. Now, AI and machine learning is a hot topic today, but since day one, I've been at NCBI NLM, which is about 18 years now. We've always interested in using AI for improving PubMed search experience. So here is a list of search assistants or features that were made exclusively by using computational methods, AI and machine learning for enhancing our user search experience. I'm not going to the detail for this talk about these features, but one thing that differs us from general search engine or commercial literature search system is that we are very transparent about how we implement these features. For each feature, we would have either a publication or a blog post about what's under the hood, how we exactly utilized AI or machine learning to implement these features, because we believe the more transparent we are, the better the users will trust and understand these features and better utilize these features. In fact, back in 2018, we wrote an article also not only showing how these features were implemented, but also how our users are responding to these features. Because as you know, once a feature is implemented in PubMed, we continuously monitoring its usage and our user response. Some of the features are getting routinely updated, while others may be retiring if something is not heavily used by our users. Obviously, my own favorite is the best match, which we developed for the new PubMed back in a few years ago, and this now is the default search compared to the previous reverse time order search in PubMed. Now, PubMed is great, but as you all know, finding papers to read is really the very beginning, the first step in scientific discovery. And especially today in an era where we have, I would say, information overload, where we have information information needed we're trying to answer, we can usually find 3 or 4 keywords to search for those in those search engines. So it's not that we can't find papers to read. The opposite is usually true. We have too many papers to read because given a set of keywords, 3 or 4 terms, we are often getting returned hundreds, thousands, or tens of thousands of papers. So for individual researchers like yourself, like ourselves, excuse me, we just don't have the time to go through those long list of papers. So the million-dollar question, or the big problem, is how can we teach computers, to read that set of papers? Because computers can really scan a large set of papers very efficiently. But we want them to able to not only to scan the papers, but trying to understand just like human scientists, and then make the connections that human scientists were never able to afford to do. So towards that goal, we made some baby steps. We teach, for example, computers to read articles and then try to understand what are the important concepts or entities in a biomedical literature, scientific publications, and what are the relationship between these entities. So in terms of entities that are important in biomedical research, I think everybody would probably agree, diseases, genes, and drugs and chemicals, these are most important concepts. And this also happens to be the most searched topics in PubMed after author searches, because everybody likes to search their favorite authors, or including themselves in PubMed in a way. So this becomes then, these entities becomes the focus of our research from the text mining or NLP perspective. We also added in genomic variants as well as species. Because, for example, if we want to extract genes or identify genes correctly, we need to know, is this a human gene versus this is a mouse gene, or bacteria. You can see here, I showed you some of the examples of each category or each entity type, where you can see that for the same entity, there could be many different names or different names referring to the same entity, and the vice versa is also true. The same entity could refer to different, same name can also be referred to the same different entities. This is because human language is very rich, but at the same time, it's very ambiguous. So if we are thinking about to recognize these entities using a dictionary lookup, I have a name of gene, a list of gene names. Can I just using that method to do a string matching? That's not likely to be successful because people have tried that in the past, and the performance is not good because, like I say, because the ambiguity issue in languages. So our solution to that is machine learning, and thanks to 2 very talented and hardworking scientists in my group, Drs. Wei and Dr. Lehman. So over the years, we have built a number of entity taggers, tools that can automatically extract disease names, gene names, species, chemical drugs, and mutations from the free text. As you can see, some of these tools have gone through multiple iterations because we continue to improve these with the latest AI techniques or AI methods. And in the past, we've gone through multiple iterations, and we started to developing tools specific for one type. Like we have genome for disease, we have genome for genes. But then back in 2023, we also developed something called all-in-one tagger, because we find that it's beneficial actually to train a one tagger that can automatically recognize all 5 different entity types. So one thing we've done is that we believe open science, so we make these taggers publicly available, including their source code, and that gets actually being used quite a bit. So for those of you who are not familiar with this topic, I just want to show you one concrete example what I mean by these gene taggers and how what they can be doing with a PubMed abstract. So in this particular abstract, I'm using example for variant or mutation recognition. So in the first step, our tagger would recognize there are three mentions, three different strings that in the article that refers to the same mutation. Okay, but in addition, we also have a gene tagger which knows that this article is about F11 gene and this mutation is related to this F11 gene. And because of that two information together, we can not only recognize these three mentions of the same mutation, but also ground this mutation or normalize this, map this mutation to a standard database identifier. And in this case, we use the NCBI dbSNP, which is a variant for SNPs and using an RSID as its standardized consistent persistent identifier. So because we make our NLP tools publicly available, we have found that there are quite a bit of download of our NLP software. And what people do with that is they apply these software sometimes to a subset, sometimes to the entire set of PubMed articles. If different groups are doing this for the for different reasons, but can we actually do this for different groups? So that was basically the idea that back in 2012 we created a PubTator surface where we applied our different taggers to the entire set of PubMed articles, abstracts, and then aggregate their results in the back end and render them in the front end web browser. So as long as someone have access to internet, they can open their browser, they can look at the annotations of a particular article and looking at what's our annotation look like for a particular type of entities and decide whether or not they would like to using this in their domain or use cases or their applications. Because I have to say, none of our taggers, as you see here, is perfect. They don't have 100% accuracy, but even you have asked humans, experts, domain experts to annotate genes for the same article, the two human scientists would not probably not be 100% agreed with each other. So, so even though we don't have the perfect accuracy, it does not mean they are not useful for other downstream tasks. And this really helps not only those, saves time and resource for those computational groups. They don't have to apply and download all the data from PubMed and apply our tools, but it also helps those groups where they maybe do not have an NLP specialist or computational scientist. They just want to extract our data, annotation data. And we provide APIs for them to do that, because if once they looked at the examples in PubTator web browser and they like the result, like the quality of our annotation, they can write a one sentence code about retrieving the data from a subset of articles of they're interested in, or they can also use an FTP to download basically every article annotations. So this is very convenient setup for our users, and turns out to be API is the real winner here, because we see that you see that in this figure, we have over 1 billion API calls over the years from a wide variety researchers from different groups. And PubTator has been utilized for a variety of reasons and a variety of different use cases. Oftentimes, people, when I make a presentation or somebody in my group make a presentation about PubTator, and then after the presentation, we would somebody would come to us say, we are a user of your tool. And then we would follow up these conversations about how the tools are being used in different use cases. Other times, people would send us an email starting with, thank you for your great service, but can you help us with this and that? Or sometimes they just alerting us that our system is not working properly. This is all good because the worst thing, I think, is that you have a surface and you never get any feedback from your intended audience. So another way we look at use cases is we look at papers who cited PubTator. So from those different sources, we categorized, there are 5 different major use cases of PubTator, from helping other similar mind NLP researchers to do information retrieval or relation extraction NLP tasks, to people like yourself, perhaps focusing on database curation, to others who actually working on bioinformatics. And during the pandemic, we also find PubTator played a role for clinical research. But obviously today I would be focusing a little bit more on the database curation part. We have been actually working very closely with this community for a number of years now. And back in 2017, 2018, for example, we worked with both UniProt, SwitchProt, as well as GeoOS Catalog. Both are NIH-funded projects. And regardless, even though they have a very different curation focus and scope, in both cases, we find that NLP can achieve 2 to 3 times higher precision and help improve productivity and data curation up to 3 times compared to a manual annotation, without any sacrifice in the end of the data quality. And because of these findings, we actually, PubTator has been utilized in the production pipeline of the data curation projects for both of these projects. And because of these different use cases, and details can be found in those papers, by the way, I'm not going into the details. Because of that, we have actually continued to develop in PubTator. As I mentioned, we started this now over 10 years ago at the very first version of PubTator. And then back in 2015, we added API, which turns out to be a huge success, because that turns out to be the most convenient way of outside users retrieving data annotations from the PubTator surface. And then 2016, for those who are interested in bulk download, we give them the FTP so that it can relieve some of the pressure on the API side. And then 2018, we actually went further, go beyond just the title and abstract, but for extracting entities and the relation and entities in the full text. And in fact, in one particular entity mutations, we even went to the supplemental materials. So that's number 2018. And 2019, because the newer AI methods in deep learning, especially transformers and BERT models, were becoming mainstream methods and showing very promising state-of-art performance. So we transitioned and switched our taggers with these models. And lastly, the latest update was in 2023, where for the first time we included relation annotations. And that's also the basis of the PubTator 3.0 version. So let me say a little bit about the relations that we are including in the PubTator system. So here is a concrete example where you see that, or a toy example, you see that in the abstract, in the title, we have a disease, Alzheimer's, we have the gene, and we have the variant. So prior to the 3.0 version of the PubTator, we would only including the annotations of 3 separate entities. But in the current model, in the current system version, we're also indicating that there's a relationship between the gene and disease as well as with the variant. Now, obviously, this is a toy example, so the relationship between these 3 entities is very obvious to you as well as to the computer. But in reality, when we have a blend raw PubMed abstract, there could be multiple diseases, multiple genes, and variants. Not to mention, sometimes we also have negations. So to tease out and figure out which gene, is related to which disease, becomes not a so trivial task. And that was basically my hesitation to including relation extraction results in PubTator, because the previous performance on relation extraction using automatic NLP methods is really a wide range between 30% to 80%, depending on the type of relations. For certain relations like PPI, protein-protein interaction, the performance may be higher, while for others may be lower, and significantly lower. And the reason for that is most of the methods, typical methods used for relation extraction rely on rule-based systems or patterns or sentence co-occurrence. If two entities, gene and disease, they are co-mentioned in the same sentence, then we believe there's a relationship. And as you know, that is not necessarily true. And oftentimes it's not the case. The better way to address this is using machine learning. And that turns out to be a more accurate method. But on the other hand, it requires a massive training data. And prior to 2021, basically there was no such a data set publicly available for a wide variety of different entity relation types. And a lot of the method developed worked well on the DDI or drug-drug interaction may not work very well on protein-protein interaction, vice versa. So looking at that gap, we really mean three postdocs, researchers in my group, finally decided that if we're going to using or including relations in PubTator, we have to develop some data set to train such a machine learning model. And that's when they spend almost a year, obviously not full time, because otherwise that would make them make people crazy, but a significant portion of their time, like 30% of their time throughout year, to annotate a very large corpus or training data for relation extraction method. And as you can see here, this data set was developed during the pandemic. It covers the 5 entities already in the PubTator system, but with these 5 different entity types, there could be a wide variety of different relations between the pairs of entities, right? And even between the same pair, for example, disease and drug, disease and a chemical. It could be a chemical is used to treat a disease. It could also be a chemical it has a side effect of causing a disease. So there could be different relation types between the same entity pairs. But nonetheless, they actually worked on this and made an annotation for that set of 600 articles initially. And later on, we added another 400, making currently a total of 1,000 PubMed abstract, fully annotated with 5 entities and I think dozens of relation types. But for the initial 600, we cover 20,000 entities, which includes 7,000 unique entities and 7,000 relations. Covers disease and gene, which is not it's quite typical actually. If we look at the scope of the PubMed literature, disease and gene certainly are very prominent topics, and then followed by the drug and chemicals, basically, drug and diseases, and then we have gene and gene for 17%, and a chemical and gene for 14%, and mutation and disease for another 14%, and the rest of them accounts for another 10%-ish. Okay. So with that data set, we call it BioRed, the biomedical relation extraction data set, we finally are able to build a state-of-the-art NLP algorithm, relation extraction algorithm, for these different entity relation types. And this is done by my current postdoc, Dr. Lai, in the group. And as you can see, he's been with me for some years now, and he has developed a number of different tools. Actually, all these tools that I listed compared were developed by him. And we started in 2020 working on this project. And the best performance at time when we used this BioRed test set was 72%. And because of the training data we developed in 2023, we were able to move that to 80%. And he just finished complete a new project where he actually leveraged some of the best method that published, and then have a very clever idea about making different relation extractions, finding them all together. And this is something that he added it with improving the relation extraction with directionality and further improving the accuracy to 82%. And this is something that is currently conditional accepted and for the ISMB meeting later this year. And then not only this method works on the BioRed data set, it also works on a separate independent data set. We call it BC5-CDR, which has only two types of relations between chemicals and diseases, and that's why it's called a CDR. And as you can see, using the BioRed training data, it's with the three methods that we compared, the trend is the general finding results are consistent. It improves from the 2020 model to the 2023 and then 2025, although you would expect that because this is not trained on this particular data set, so the performance is slightly lower compared to the performance on the BioRed test set. So thanks to Boting and in his work, we eventually are able to including relation extraction results in the PubTator 3 project back in 2023. So we not only have the entities, but we also have the relations, and that to us is a major improvement compared to the previous version of the PubTator system. And because of that, we're happy to report there are even new users that are integrating PubTator into their curation pipeline. So we just learned that CTD, the database of comparative taxonomogenomic database, they recently published a paper in the Journal Database, and they put reporting their work on integrating PubTator into the manual curation workflow. After a very rigorous evaluation, they actually, and which was done completely independently from us. They evaluated, they have a targeted list of requirements that CTD needs for integrating an AI or NLP system. They also have done a qualitative assessment of the quality of the annotations of automatic methods, and eventually they decided that PubTator is the best choice out there for assisting their manual curation process. And CTD is a natural fit for this because they centers on annotating genes, diseases, and chemicals. And these are the entities we actually already, like I say, annotated, as well as their relations. So it's a very natural fit for them. But there are relations in other databases, maybe like yourselves, that are not necessarily have been captured already in the PubTator systems. So in those certain cases, what we do is we would collaborate with the domain databases from scratch. So one example is the RIA database that focus on annotating enzyme reactions. And we don't have that in the PubTator system. And what they like to do is to find out and prioritize what are the empty enzyme relations they need to curate by human curators. So we worked with their team, Dr. Alan Bridge and his colleagues, to basically set up a pipeline from scratch using the tools that we have previously developed. So we take the entire set of abstract in PubMed, and then using the literature triage tool called the ListSuggest from us, and then the entity recognition tool, all-in-one NER, and the BioRex, the relation extraction tool, we are able to actually extract all the enzyme relations from the entire PubMed articles. And then we were able to generate the figure down below where we see that there are different nodes and the different colors. the red ones represents the enzyme relations that are annotated, but only in the real database, while the blue ones are the ones that are only found in the literature but not yet annotated. And then the color yellow means they are found in both places. And then we can, if we zoom out on some of the key clusters where we see there's a lot of blue dots, which means they have only found in the literature but not in their current database, and this becomes the priority for their next annotations, basically. And all the details, again, is reported in a paper in Scientific Data last year. So now I would move a little bit, because all I'm talking about here using the tools that were mostly based on transformers and BERT model if you are NLP researcher. So these are the AI methods prior to the launch of ChatGPT. I have to say, in my 20 years of AI research, we've never seen anything like ChatGPT that is so amazing in terms of its capability for language generation and language understanding. So it's no wonder why it makes me no surprise to me that it grabbed 1 million users in 5 days compared to a much longer time for some other very popular online applications and services. Some of the early success of ChatGPT, including chatbot, including that when it's launched, it already is able to achieve a score of 60 on a USMLE medical exam. This is the medical exam test for medical students in the US, and in the format of multiple choices questions. Okay, and within less than 6 months, the ChatGPT bot is able to basically achieve a performance of in the 80s, which is just below the attending physician level. I'm pretty sure now the performance is probably already over 90. Okay, and we are so used to this kind of amazing performance of chatbot, but let me say this, that prior to ChatGPT, a lot of researchers worked on the same problem. They always trying to develop AI method that can answer medical questions, medical exam questions correctly. And a lot of efforts have put in prior to 2022, and the best performance was in the 30s and 40s. And if we, at the time, if one can improve performance by 1 to 2%, that considered very significant. So nobody, well, at least to me, I did not expect that we would basically solve this problem in such a manner back in 2022. So after seeing this type of amazing results, so the natural question in 2023 at the time for us is, now should we change our taggers using, right? So that's very natural for us to think about. Instead of using the previous generation transformers or BERT models that perform the best at the time, should we using ChatGPT for the task? So in 2023, we actually started working on a benchmarking study where we set out looking at 6 different NLP tasks, including relation extraction, including named entity recognition, question answering, that are important in biomedical NLP research. And for each task, we look at 2 different data. We use 2 different public data sets. For example, for NER, we used NCBI disease as well as BC5 CDR. And then we look at both open source as well as proprietary large language models, including the GPTs and the llamas. As you know, you can actually use these large language models in different settings. So we used zero shot, we also used few shot, and fine-tuning different settings to see whether or not we can get the best performance under these different settings. So not going to the details about the paper, I mentioned the paper, we studied the research in 2023, and we submitted our initial version in late 2023, and then we gone through multiple iterations of revisions for this paper and added additional results. Finally, our paper is now will be published this month, I believe. But the main findings are in this table 2 of the paper, where we show that on the left side, the first 2 columns are the tasks. So for example, we have the name entity recognition task, we have the relation extraction task, and then for under each task, we have 2 different data sets. For the NER, we have the BC5-CDR and NCBI disease. And then the next to that, the second column is the evaluation matrix. So we used F1 score, the balanced recall and the precision. And the third column that I highlighted in the blue are basically the best performance prior to the large language model, prior to those foundation models. So for example, using the PubMed BERT model, the best performance is 0.95 for BC5-CDR and 0.9 for NCBI disease entity recognition. For relation extraction, it's lower in the 70s, 73 using BioBERT for chemical protein relations and 0.79 using BioBERT for drug-drug interaction. And then the rest of the table on the right side are basically the performance using different large language model foundation models. We looked at GPT 3.5, we looked at GPT 4 and the Llama, and we also looked at under the 0 shot, meaning no examples, 1 shot, 1 example, 5 shot, 5 examples, as well as fine-tuning using the training data for fine-tuned the methods. So as you can see in this table, except for the question answering tasks where the best performance comes out those foundation models, or other tasks, in fact, the best performance is still coming from the previous generation BERT models. So as a conclusion, we find that for information extraction, which is the most relevant task for data curation, using 0 shot or few shot with those foundation model is less recommended as part of the findings and the results or takeaway message from this particular study. Instead, the top choice remains using fine-tuning the BERT models. If we want to have a accurate relation extraction results for particular relation type, perhaps using the BERT model with the fine-tuning is still the best way to go, at least for the moment. Now, obviously, this research is advancing very quickly, and we've seen evidence now actually using fine-tuned foundation model is also approaching very good results, and this is something that we're working on right now. We also did along this study is a detailed error analysis. We looked at why these foundation models make mistakes. We find there are issues in consistent results. Sometimes they make mistakes in returning missing proper formats or proper results. But one big issue that we find that is new, relatively new for the AI model is hallucination. So this is an issue known not only for this type of tasks, but in general for the chatbot, the large language models, that they tend to generate fabricate incorrect information, but in a very confident manner. So the final 10 minutes, I'd like to show you the two-way street. So far I always talked about how you can utilizing AI for assistant data curation, but I also, in the final 10 minutes, I want to show you a study we just completed where we showed that actually curated knowledge, thanks to you and this community, can really help improving and addressing reduced hallucination. And the example I'm going to show you is through a case study of a task called gene set analysis. Perhaps this is also very not new to you, because this is really a very common task in biomedical research or bioinformatics research, because oftentimes people doing high throughput experiment, they're generating a list of genes, and these genes can be differentially expressed or under different conditions, and so on and so forth. And the goal to this process or this task is to figuring out what would be the collective function of that set of genes. Okay, so the go-to method people use these days is gene set enrichment analysis or pathway analysis. Basically, they're comparing their input, their list of genes to some of the curated databases of that subset. Maybe there's overlapping with some of the non-curated gene sets. And there are two main, and that works okay, but there are two major limitations. One is that it's limited to what has been curated in the databases. Again, if your gene list is very foreign or have very little overlap with what's in the database, you're less likely to get a good answer. Number two is that even when you get a good answer, you don't know necessarily what's the biological process underlying explaining the collective function. Okay, so people have been very creative. There has been studies to show that instead why don't you just give that set of genes to large language models and ask it to generate the set of functions. And that turns out to be actually quite successful in this paper published last year, where Hu and the colleagues showed that given a set of genes to the large language model like GPT-4, they actually benchmarked a bunch of different large language models. They can actually generate results very accurately, very comparable, even higher than the traditional enrichment analysis. But they also find that about 20 or 25% of a quarter of the issues are hallucinations. And this is, like I say, a natural problem, an inherent problem of those large language models. So the idea we had in our project is maybe we should be using those gene-centric databases, okay? Because I'm pretty sure, PubMed has already been utilized somewhat during the large language model training process. But I'm not less sure about these structured databases. They're less likely to be utilized already by these large language. In other words, the knowledge that human actually curated are probably not yet reflected in these large language models. So our idea is that we can get the we can use in the same step as the first stage, given a set of genes as the input, ask the first large language model to generate the function and generate the explanation. But instead of taking that as the final answer, we have an additional verification step. Now, obviously, you can have humans, you can recruit a human or invite a human expert to do the verification. But as you know, this will not be realistic because it cannot be scalable, this process. You can also ask another large language model to do the verification of the results of the first large language model. And people have done that. It has some success, but it's very limited because research have shown doing this, having a second large language model to verify the results of the first large language model is highly biased. So our idea is that we're still going to do the second round using an automatic large language model to verify the results of first large language model, but we would not let them do this verification freely, but rather we would require them to check and using curated gene information in the domain databases. So this is we call it AI agent for self-verification. We do this, but we ground this knowledge verification process completely on curated knowledge in domain databases. So here is a more concrete example where, as you can see, for example, the first large language model will generate a statement that these 5 genes, ERBB1, ERBB2, ERBB4, so on and so forth, is involved in RTK signaling. Is this correct? Well, this has to be verified automatically. And then how do we do this? Well, basically, given this set of genes, we would activate these different APIs to look information about these genes or subsets of these genes in the domain databases. And once we gather their information, then we put that back to the second large language model and ask it to verify the claim or the statement in this RTK signaling. And in this particular case that I'm showing you here, in fact, the large language model, given the information from the domain database, it does not think it's correct. Instead, it should be MAPK signaling pathway instead of RTK. So just by doing this, I'm showing you here in this slide that on 3 different benchmarks, the gene agent with this self-replication agent based on domain databases showing dark blue color is able to outperform the standard GPT-4 consistently and sometimes by a very large margin. And even more, we actually tested this on 2 different, some novel gene set has never been published based on the experiments done by my colleagues at NCI. So we take 7 gene lists that are never published before and from mouse melanoma cell lines, and then we generate results based on standard GPT-4 as well as gene agent, which basically is standard GPT-4 plus this self-verification module, and ask 2 domain experts to blindly rank which one is better. So in this slide, we show you that in 5 of the 7 cases, the gene agent is able to perform better based on the 2 domain experts. Is a little bit better. So this is basically to summarize the type of research we're currently doing with large language models. We are very interested in evaluating standard large language model on different tasks. Some tasks are related to biomedical research and data curation, while others are more related to clinical research. And then throughout the way, because we as a academic lab or a research lab, we just don't have the resource to compete with the private sector to produce the best and the most powerful large language models. But our thinking philosophy is that we can always improve these standard large language models with some technical improvement, and using the knowledge and using the curated databases is certainly one way to go. And this could not happen without the extraordinary efforts of the data curation community. So thank you for doing that. Before I close, I just also want to very quickly mention there will be a new NLP challenge for data curation related to AI called a BioCreative challenge. And we are one of the BioCreative, for those who don't know, is a community-wide challenge event for evaluating biomedical NLP systems. Has been running since 2004, so over 20 years, and this is the 9th version of the editing version of the BioCreative. So if you're interested, if you happen to be one of those our colleagues and peers in doing the NLP research, you may want to take a look at the BioCreative 9 challenge. So thank you for your attention. Even though I'm presenting today, I'm really presenting on behalf of my wonderful team as well as our collaborators both within and outside of NIH. So I hope that you find this work interesting